I'm a scientist by training. My background's in genetics and neurobiology. For a bit over 20 years now, I've focused exclusively on the use of genetic medicines for treating disease. During that time, I've become very familiar with the existing delivery modalities, whether they're viral or non-viral. And I've also become familiar with where they are successful and where they're severely challenged. I was introduced to my SonoThera co-founders, Steve Feinstein and Michael Davidson, in 2021. Steve and Michael shared with me some really interesting proof of concept data that showed that ultrasound could be used to selectively deliver a genetic payload to virtually any organ system in the body. And as you can imagine, since I came at this from experience with all the problems in delivery, I got very excited about the potential of this technology platform. All of these approved gene therapies rely on using adeno-associated virus, otherwise known as AAV, as the delivery vehicle. With that, there are certain challenges that are associated with it. Now, the field is really quite interested in shifting genetic medicines not only to rare orphan diseases, but expanding into much more prevalent diseases such as heart disease and obesity and diabetes and Alzheimer's. So there are really 5 key limitations: organ biodistribution, redosing, payload constraints, safety, and patient access. Number 1, organ biodistribution. The existing delivery modalities are limited to the retina, the spinal cord, the liver, and muscle. If you consider non-viral approaches such as lipid nanoparticles, they are also limited to the liver and, in some cases, the lungs. So organ biodistribution is currently a major limitation of existing modalities. Number 2 is redosability. So the concept of a one-and-done therapy that lasts a patient's lifetime is great in principle. But in real-world practice, the durability of these therapies is actually somewhat uncertain. So the effect on that is that patients and physicians have been somewhat reluctant to adopt these therapies. And the underlying problem is that the immune system recognizes these viral vectors such that re-administration of the virus is really impossible because it rapidly gets inactivated. So that precludes the ability to do redosing over time. LNPs aren't much better because upon redosing, they drive an inflammatory immune response. So there are no approved products using LNPs for gene therapy. So the third constraint I mentioned is the payload limitations. So when using AAV, one is limited to a relatively small payload capacity of 4.7 kilobases of DNA. When using lipid nanoparticles, one is also limited in size, but you're also limited in the format of nucleic acid. DNA delivered by LNPs drives a major inflammatory response. Therefore, most attempts have really focused on delivering RNA with lipid nanoparticles. The fourth challenge is really on safety. It amounts to inflammation, immunogenicity, and a really poor on-target versus off-target profile when you look at different organ systems. And the fifth challenge is the patient access. AAV manufacturing is quite complex and costly, and that in part drives very high product costs. These range typically in the $2 to $3 million price tag for these therapies, which has really limited patient access. And when you think about it, it's really only relevant to rare diseases in first world countries. It leaves many patients and diseases untreatable. SonoThera's platform consists of 3 components. There's the genetic payload, there's acoustic microspheres, and 3, there's acoustic energy. We do an intravenous infusion of the genetic payload along with the acoustic microspheres. Those 2 components flow throughout systemic circulation, and they can access virtually any organ system in the body. We then use the acoustic energy and direct it only to the target organ of interest, and that activates the acoustic microspheres and drives several mechanisms of action. Most interestingly, it creates microscopic pores to form in the cell membrane of target cells that allows for our genetic payload to flow through the cytosol and transit across the nuclear envelope to eventually drive therapeutic gene expression to occur in those target cells. So there are several advantages of this. We can selectively target nearly any organ system in the body using essentially the same platform with minor modifications. So we can effectively redose without immunogenicity or toxicity many times, and that allows us to titrate the dose to the appropriate therapeutic level over time. Payload constraints are nearly non-existent for our platform, which means we can deliver very large payloads. We can also deliver a variety of different nucleic acid formats, such as DNA or RNA, or combinations of DNA and RNA. So as we think about more complex genetic medicines, such as gene editing, which may require the use of a guide and a nuclease encoded as RNA, along with a DNA donor, those can all be combined together with this platform and effectively delivered to an organ system. Our ultrasound acoustic microspheres are all FDA-approved agents approved for diagnostic imaging. And our ultrasound hardware and probes are all FDA cleared as well for diagnostic imaging. So with that comes a very stellar safety track record of decades of safe use in humans. The mission of SonoThera is to develop an ultrasound-guided, non-viral gene therapy platform that can be used to provide patients with the next generation of safe and effective genetic medicines.